So this is chapter 4 on how the cells obtain energy. So here we start out with an example of a hummingbird, and we all know hummingbirds spend most of their time in flight, and they can fly an incredibly long distance for their size. So this bird is able to maintain its energy by taking in food, and it transforms the energy contained in the bonds of the food molecule into its power to fly itself around, and it does this through a series of biochemical reactions. Ultimately, if we look at it, everything is going to get its energy from the sun. So although you and I do not directly do photosynthesis to get our energy from the sun, we do consume plants that use photosynthesis to capture sunlight, and other animals that have eaten plants, they have gotten their energy from the sunlight. So what we're really looking at is the first, or just one half of the carbon cycle here, in how we use carbon from the plant matter and release it as carbon dioxide. So we as carnivores are going to eat the herbivores and eventually decompose the eventual decomposition of the plant and the animal matter will contribute back to the nutrient pool. So as we decompose, when we die, we actually will go back into feeding the rest of the world, releasing our nutrients back into the cycle. But everything does originally start out getting its energy from the sun. So with metabolic pathways, we have two categories they fall into. The anabolic pathways are going to be the ones where you have smaller molecules and they'll be built or assembled into larger ones. So here we see it starting out with 4 simple green balls. When we put energy into it, we can attach those 4 green balls together. A catabolic reaction is going to be one that breaks something down. So these 4 green balls start out attached. When we break the bonds between them, it's also going to release energy into the environment. Both of these types of pathways are required in cells to maintain their energy balance. So these are some examples of energy that gets transferred and transformed from one system to another. The food we consume is going to provide our cells with energy, and we use that energy to carry out our normal body functions, just like the sunlight is going to provide the plant with the energy it needs to create its chemical energy. So it starts out showing ice cream here as chemical energy going into these kids that will give them the kinetic energy of moving their bike around. Over here it's showing sunlight that will provide the energy to make chemical energy in the form of carbohydrates that are in these leaves. So when we're looking at potential energy versus kinetic energy, potential energy is going to be energy due to location. Here in this dam, there is a lot of potential energy because this water could potentially fall down. Over here in the waterfall, you've got a lot of kinetic energy. The water is falling down. It's in the motion of actually falling down. So we also have endergonic and exergonic processes. An endergonic process is one that requires energy be put in. Ender means inside. The exergonic process is going to be one that releases energy. I remember that with the EX being for exit. So here you've got an endergonic process of putting the energy in to make the chickens and then releasing it as it's rolling down the hill, here for these balls rolling down the hill. In order for a reaction to take place, it's going to have to go from one energy level to another energy level. Here it's showing a reaction pathway, the amount of energy, and what enzymes do. So here you've got the reactants at a lower level, the products are at a higher level. In order for these reactants to become products, you're going to have to put some energy into them. This difference in here, represented by this little triangle we refer to as delta G, is how much energy you have to put in. Sometimes you've got to put in a little bit of extra energy to get over the hump to get things started. And that's what you're seeing with the purple line. That's without an enzyme. What an enzyme is going to do is it's going to lower this activation energy, this extra little bit you have to put in to get things started so that it's not as big of an obstacle to get over. So here with the enzyme, you can see it's a smaller hump that you have to get over. So that's what enzymes do, is they lower the activation energy in a reaction, but they don't actually change the amount of energy that is required to go from reactants to products. Enzymes are going to change shape as they work. You have your substrate here, and then you have the active site of your enzyme. And we say it's an induced fit model. So what will happen is the substrate will go into the active site. And when it does, it's going to change shape just slightly. So we say it's an induced fit because it will adjust to the lock and key mechanism. Then you have your enzyme product complex. The products will leave the active site and your products will be released into the environment. Enzymes can also be regulated, and one way of regulating them is allosterically. So with allosteric inhibition, this works by indirectly inducing a change in shape in the active site so that the substrate no longer fits. This is the example here on the left. You've got the enzyme, you've got your allosteric site, and your substrate. So when this inhibitor comes in and binds this allosteric site, you are going to change the active site shape so that the substrate will no longer fit, so that the enzyme cannot be used. That's an example of allosteric inhibition. With allosteric activation, when that activator is going to come in and bind that allosteric site, what will happen is the active site is going to change shape so that the substrate can actually fit to it. This is referred to as allosteric activation. So looking at chemical reactions is an important way of how we've managed to understand how pharmaceutical drugs can be used to benefit us. So one of the important things is if you're looking to manufacture pharmaceutical drugs, you do have to understand those metabolic pathways. Metabolic pathways are just a series of reactions, and they get catalyzed by multiple enzymes. With feedback inhibition, there's going to be an end product of the pathway that's going to inhibit the upstream process and act as an important regulatory mechanism. So here we have our inhibition of the process. So normally you would have your substrate would go in and bind to the enzyme, and it's going to make its first intermediate substrate and its second intermediate substrate, and finally its end product. When the end product starts to build up, with this type of inhibition, what will actually happen is the end product will come around and it will inactivate the enzyme or inhibit the enzyme from making any more so that you don't continue to build up the product. The currency we use to move energy on in animal cells is called ATP, or adenosine triphosphate. This is an example of what this looks like. So you've got a ribose attached to an adenine, and then you've got three phosphate groups here, an alpha, beta, and gamma phosphate group. Where this molecule holds a lot of energy is in this bond holding on this third phosphate group. It's called ATP, or adenosine triphosphate, because of the three phosphates. If we were to take one of those off, it would be ADP, or adenosine diphosphate, only having two phosphate groups. So the first process we use in order to make energy from the carbohydrates that we consume, like glucose, is glycolysis. It literally means to lyse glucose. Here you start out with a glucose molecule. It's got 6 carbons in it. You're going to put some energy into the process in the form of ATP. 2 ATPs are going to come in. They're going to take off the phosphates, and you'll end up with 2 ADPs as a result. It's going to put it onto the glucose, and it's going to rearrange itself to make fructose diphosphate. Sometimes this is referred to as an energy investment phase because you're having to put some energy in to get the reaction started. enough energy is put in that these are split in half, that we have 2 of these glyceraldehyde 3-phosphate molecules. You can see all the carbons are still accounted for. You had 6 up here. Now you have 3 on this side and 3 on this side. Your 2 phosphates are still accounted for. We're going to go into the energy production phase here. We're also going to move some electrons around. This NAD plus will accept some electrons and become NADH. Also in the process, you're going to take ADP here, 2 of them, and you're going to pull off phosphates from this glyceraldehyde 3-phosphate as it progresses through here. The phosphates come off, they go on to ADP to become ATP, and you're left behind with 3 carbons in the pyruvate. We have 2 pyruvates here, so all of those carbons are still accounted for. So another picture I like to use for this, right here with glycolysis, sometimes a little bit more detail is a little bit easier. You can actually download this picture here below the document that you found the video in. We start out glycolysis with glucose. Each of the little black balls on here represents a carbon. So we have 6 carbons. When we put in the first ATP, we're going to take the phosphate off, and the phosphate's going to be represented by this little red ball here being attached. It's on carbon number 6, so we call it glucose 6-phosphate. That's the first ATP we've put in. We do put in 2 to get this process started. So the second one comes in, and it's going to put a phosphate on the other end. Now we have fructose 1,6-bisphosphate. What that big mouthful of a name actually means is that you've got fructose, now is the sugar, and on carbon 1 and carbon 6, you've got a phosphate. And because there's 2 of them, we have the bi in front. So at this point, you'll have put in enough energy that you split this in half. We're going to move the electrons around, and this is where you're seeing the NAD plus come in, that will end up taking some electrons and being released as NADH. We're going to have the energy payoff phase here, where you're going to take one of these phosphates off, and you're going to put it on the ADP so that it will end up making ATP. What you're left with still has the 3 carbons here. You have the phosphate group, and we're going to rearrange that a little bit. Here you're just going to call it 3-phosphoglycerate or 3-phosphoglyceric acid. You'll rearrange it to be 2-phosphoglyceric acid. It's just moved on to carbon number 2. It rearranges a little bit more. We've got phospholinopyruvate, and we're finally going to pull off that last phosphate, put it on this ADP, and it will make ATP. So in the next step, we have that pyruvate or pyruvic acid here. We have those 3 carbons. It's going to be transported into the mitochondria. As it does that, you're going to have coenzyme A come in, and you're also going to have NAD plus come in. It's going to take an electron and move it, and you'll end up with NADH. When the coenzyme comes in, you're also going to remove a carbon, as well as 2 oxygens. This will form carbon dioxide. This is where part of the carbon dioxide comes from that you exhale, is you are breaking it off of the 3 carbon chain of the pyruvic acid. When you pull that off, you'll be left with the 2 carbon acetyl-CoA. The CoA came from the coenzyme A here. This is going to feed into what we call the Krebs cycle or citric acid cycle. It's also sometimes called the Tca cycle. The acetyl-CoA will come in. It's going to bring 2 carbons with it. As it goes through a series of steps breaking it down, you're going to pull off those 2 carbons. They'll combine with oxygen and be exhaled as carbon dioxide. This process is aerobic in that it requires oxygen to take place, because if you don't have oxygen to attach to these 2 carbons, the cycle is no longer going to progress. As we do that, we're going to move some electrons around. we're going to have 3 NAD plus that we will place electrons on and end up with 3 NADH, as well as FAD take on some electrons and become FADH2. We will also take a phosphate and put it on the ADP to make ATP in this part of the cycle. This here again is another picture I like to use. We're starting out with our 3 carbons here of the pyruvate or pyruvic acid. You're going to pull off one of the carbons is carbon dioxide here. 2 of them are going to enter in. They're going to attach to these 4 carbons that were left behind from the previous turn of this cycle. We'll have these 4 carbons plus 2 of these carbons that come in here. That will make 6 carbons. These 6 carbons are going to rearrange a little bit, and you're also going to pull off a carbon, combine it with 2 oxygens to release carbon dioxide. You're going to do that a second time to release more carbon dioxide. As you do that, electrons are going to be released, and they will be transported on the NAD plus that will become NADH. As we rearrange these 4 carbons here, they're also going to release some energy and a phosphate that will attach to ADP to make ATP. These 4 carbons continue to rearrange, moving more electrons with the FADH2 carrying them, and finally coming back to the beginning when you have your 4 carbons that you started out with. So next is the electron transport chain. This is going to occur inside of the mitochondria, and it's a set of molecules that are going to pass electrons down through a membrane, and it supports a series of oxidation reduction reactions. Just in simple terms, oxidation reduction reactions involve moving electrons. At the very end, you have ATP synthase. It's an enzyme complex that's going to use the hydrogen ion gradient to regenerate ATP from ADP. So here, as they're being passed along these protein carriers in the membrane, what's going to happen is the electrons from NAD plus will be dropped off here to NADH. Each time you have one pass through one of these, you're going to have it pump a hydrogen ion to the other side of the membrane. That's what's going to create this gradient of having all of these hydrogen ions on the one side of the membrane that want to get to the other side. So when you allow those hydrogen ions to move through to the other side through the ATP synthase, the enzyme is going to help put the phosphate on ADP so you can have ATP. So I also have another picture that shows an electron transport chain slightly larger. So here you have your NAD plus that has dropped off the electron. It's going to move up, get passed along the chain. Here it's showing the hydrogen ions being pumped onto the other side. FADH2 is going to do the same thing, except it's going to cut in line a little bit. What ends up happening is you have more hydrogen ions that are pumped from NADH than you do from FADH2. And then down at the end here, coming through the ATP synthase. So it's similar to a battery where you have positives and negatives separated, and when they want to come together, they're willing to do some work for us in coming together. The work they're going to do is to put the 3rd phosphate on ADP so that we can have ATP. Lactic acid fermentation. This is something you've probably heard of with exercising. When muscles become exhausted or you are outside of the aerobic range of activity, they're going to produce lactic acid as a byproduct. It's done in fermentation. You do this when there is not adequate oxygen available to keep going through with aerobic respiration. So we start out with glucose, it goes through glycolysis to make pyruvate. When you're doing fermentation, that pyruvate is going to get converted to lactate. During glycolysis, you have NAD+, two NAD pluses that will be converted into two NADHs. In order to keep this going, you need to have a supply of NAD+. This is where your source of ATP is. In fermentation, you're going to take NADH and turn it back into NAD+. This will replenish the supply of NAD+ so that you can continue to do glycolysis and generate energy. So there's two common forms of fermentation that we look at in biology. We take pyruvate to make acetaldehyde, and the acetaldehyde will then make ethanol again. It's going to replenish the NAD+ supply. So that's what happens with alcohol fermentation. This is what is done in wine production, so fermentation of the grape juice to make wine. It also produces carbon dioxide as a byproduct. These tanks have valves so that the pressure can be released, but otherwise it would build up pressure. In mussels, you're going to just make the lactic acid from there. No carbon dioxide is produced with lactic acid fermentation. So there is other types of metabolism that occurs. This green color is seen here in the eruption of hydrogen sulfide. Here we have anaerobic sulfate reducing bacteria. What they're going to do is release hydrogen sulfide gas as they decompose the algae in the water. So there are other metabolic plants out there. So we do eat things other than just glucose. Any of the other carbohydrates that you would consume are going to enter into the pathway in glycolysis. Some of the amino acids will actually enter in up at this pathway. So if you consume proteins, some of them will enter here. Some proteins actually enter a little bit further down the line in the pyruvate oxidation, and some will come in the citric acid cycle. When you consume fats, they are going to have a glycerol on them. That glycerol will come into glycolysis. Your fatty acids are going to come in at pyruvate oxidation and then proceed through the cycle in the citric acid cycle. And then you have some amino acids that come in later in the citric acid cycle. So if you have glycogen from your liver and muscles, that will actually come in here in the carbohydrates.